molecule called CXCL10. A debt of gratitude to Julie Ho in Winnipeg, who has done a great deal of work on this molecule and who got me interested in doing a pediatric version of her adult study really many years ago. This is a cytokine or a chemokine, I should say, that's induced by interferon gamma, which is in the acute inflammatory response. It's secreted by a number of cells within the kidney and induces white blood cells to move towards the area of inflammation that is being signaled. And for that reason is associated with acute rejection, but is also associated with other forms of inflammation. And when we looked at this in a multi-center study across Canada, we were able to identify that there is a significant association with acute rejection, even in the setting of subclinical rejection, which means that there wasn't any change in the kidney function at the time that we did the biopsy. So this is by definition better than serum creatinine at picking up rejection. The other thing that we noticed for the study was that the level of inflammation, if you look at the average levels of CXCL10 over the first year after transplant, there were really sort of three distinct groups that we could characterize. One with very low inflammation, who basically, if you look at the right-hand side of the panel, had very little change in their kidney function over the first year. There's a middle group that had a modest decline in kidney function, but then there's that top group that had a quite substantial decline in kidney function over the first group. And this suggests that inflammation over the course of the first year in general, whether it be because of rejection or because of BK nephropathy or some other reason, is quite deleterious to the graft function. So even in the absence of identifying rejection, if we are identifying chronic inflammation in our transplant recipients, it is really important to do something about it, to figure out why they have inflammation and to treat it. So this study that we're doing is modeled after a landmark study that was done by David Rush in Winnipeg across the country, which actually evaluated and demonstrated the value of surveillance biopsies in the first place. And in that study, they compared a group of patients who were getting surveillance biopsies with a group of patients who weren't getting surveillance biopsies. And perhaps not surprisingly, they identified a lot more rejection episodes amongst the patients who had surveillance biopsies. But despite that, over a 2-year period, those patients who had identified rejection did a lot better, presumably because those rejection episodes were then effectively treated and that they did not develop more chronic forms of inflammation as a result. So we did a feasibility project first at BC Children's Hospital, and the first was to just reaffirm the diagnostic thresholds that we had for rejection, which we were quite able to do. And you can see the panel on the right is patients with rejection, BANF1A, who have substantially higher CXCL10 levels than those who did not have rejection. I'll note that the borderline group, which is a true form of rejection, it is less sensitive at picking that up. So this is not going to pick up all forms of rejection, but it will pick up those that are more severe. This is what's called a receiver operating characteristic curve, and this really just sort of shows the sensitivity and specificity of the molecule using different cutoff thresholds. So it's not that you get above a certain level and everything is high and then nothing is low. It varies. And so this is something that you can actually potentially track over time, looking at changes in the level over time to determine whether there is more or less inflammation as we go. Very similar actually to how we use measures of kidney function. And this is looking at what happens before and after you treat an episode of rejection compared to what happens when you look at the biopsy 2 months later. And we can see that in patients who are treated, those patients whose biopsies look better have lower CXCL10 levels, and those patients whose biopsies look worse have higher CXCL10 levels. I will note that nearly half of these patients still had rejection on their follow-up biopsy, which fits with what we know from previous studies. But this gives us good evidence that the trend of CXCL10 over time is a very useful indicator for clinical monitoring. We did also see two things. One is that we saw surprisingly higher levels with other viral infections, CXCL10 not so much so, but in particular BK viremia, even without nephropathy on the biopsy, the CXCL10 levels are much, much higher than what we actually see with rejection. And it just tells you that there's a lot of inflammation. Even when you don't see viral particles in the kidney, there probably is a lot of graft inflammation going on. But then when we looked at those patients who had high levels and then looked over time, We know that patients who've had these viremic episodes are at a higher risk of developing rejection later because their allograft has been inflamed. That signals your immune system and increases trafficking of immune cells within the allograft. And we saw that those patients who had sustained high levels of CXCL10 after their viral illness had resolved, they were at a much higher risk of having rejection subsequently on a biopsy than those patients whose CXCL10 came back down down to the normal level after their viral infection had resolved itself. And then the last thing we looked at in this study was sort of a one-step versus a two-step approach, very similar again to how we would manage somebody in the clinic. If you come into the clinic and your kidney function is off, we're going to look at a few things, and ultimately we'll probably just repeat the kidney function again in a few days or in a week to make sure that this wasn't just a transient change, and if it's persistent, that would then indicate us doing a biopsy. And when we modeled this, we saw that the CXCL10, when it was high in the first place, we were able to identify about 45% of the time that there was an alternative cause for the elevated CXCL10 such that we didn't need to do a biopsy, which would leave us doing a lot of biopsies and having a positive biopsy result only in about 24% of cases, which is frankly still better than what happens with most surveillance biopsies. But that's a pretty low yield if we're going to be biopsying people based on the results of this type of test. But if we did a second test 2 weeks to 2 months later, and this was all retrospective modeling, we saw that actually we were able to identify in the vast majority of cases why the CXCL10 was elevated. And in those cases where we couldn't identify an alternative cause, the yield on a biopsy was upwards of 70%, meaning that most of the time we were able to identify when rejection was going to be there based on the persistence of elevated CXCL10 in the absence of another result. All right, so that brings us to the study that we've proposed. So I won't read the big long name for the study that we had to put on the grant proposal, but this is basically it's a pilot study for implementation of CXCL10 in the clinic. And the goals of the study are, as a pilot study, the goal is to establish feasibility of doing a much larger study. So this is a feasibility study, not a definitive study. And the goal is to randomize and allocate 60 patients into the study to identify and resolve challenges in executing the study protocol, including the testing procedures, and to compare the costs and logistical aspects needed to support the trial, in particular whether to do central versus local testing with the program. So this is it's a randomized control design. So there's two groups. One is the intervention group, which is the group that's going to have active monitoring. And basically this is urine monitoring that gets built into every clinic visit that they come to in the clinic, and a decision process for the clinicians to use when they get the results back in order to help guide their response to the results. And the control group, which is basically just collecting similar samples, but they go into a biorepository and they're tested retrospectively much later and not used as part of their clinical management. The follow-up period is going to be for 24 months. And the direction to indicate a biopsy. This study is not directing treatment for rejection. This study is directing indication for a biopsy to check to see whether there is rejection. Then, of course, if there is rejection, then you would treat it. We also have agreement from all the studies to be doing follow-up biopsies when they do treat rejection. So they'll be doing follow-up biopsies between 2 and 3 months after treatment of rejection to confirm and ensure that rejection has indeed resolved. The inclusion criteria are fairly straightforward. We're looking for people who are beyond the first 6 months of transplant and who are prevalent pediatric kidney transplant recipients. There's a number of exclusion criteria which are really just meant to ensure that we're not enrolling somebody who's in the midst of being actively treated for rejection, who can deliver the urine samples, who can go ahead with a biopsy, and who aren't already living with a failing allograft, such that we wouldn't be able to measure the outcome of the study. For the definitive trial, the outcomes are related to change in kidney function over time. So this is a similar design as was proposed in the original study by David Rush on surveillance biopsies. And there's a number of secondary outcome measures which have to do with the efficacy of the test and the appearance of other indicators of chronic allograft rejection. For the pilot study, for the pilot trial, this is really all about feasibility. So the first and most important outcome is the ability to actually enroll 60 patients into the study within a defined period of time, and to complete follow-up for all of those patients for the 24 months of the follow-up period. And we're going to be looking at a number of factors that relate to study refusal and participation, and our ability to do the testing and report the results in a timely way, since this testing is done integrated within the clinical labs, but this is a novel test for these clinical labs. And so we have to make sure that we can actually result those tests properly, and ensure that the validity of the testing is maintained at a high level. So this was funded this spring in the spring competition. We're in the process of completing the validation of the platform we're using, which is called the Ella Simpleplex, and we're expecting to have that done hopefully by the end of this month. The enrollment for the study is going to take place hopefully over a 4-month period. We'd like to enroll everybody actually as a block, so that we can follow them for that 24-month period as a large cohort, and that has to do with how we're going to be doing the testing, and the fiscal constraints of having enough kits and not running kits 2/3 empty. And so the trial will continue for July until June 2026, and at that time we'll hopefully analyze the results. So the piece that is very important to us is for the participants in the study to understand why they're part of the study, what they have potentially to gain from the study, what are the risks related to the study, and to keep them engaged for a 24-month period, and to make sure that that experience for them is both rewarding, satisfying, and hopefully beneficial to their care. And we've had some discussions about recruitment and information about that, and Demitra is going to talk about that in just a second. But we're also very interested in how to sustain the trial over time and ensure that participants are engaged during the course of the whole study. And then there's the knowledge translation piece, and an understanding of what the patient's experience was doing this kind of a study, so that we can learn better how to do this for the next grant, which hopefully we would be able to submit in a couple of years. So with that, I'm going to pass it over to Demitra.
Hey. Hi, everyone. My name is Demitra. For those of you that don't know me, I am the research manager with CDTRP, and I'm currently managing the CXCL10 implementation pilot trial with Tom. So I'm here to just discuss a very valuable discussion that was facilitated by CDTRP last year with 23 patient partners. And it was really to explore biomarkers and in a patient-centered focus. And the overall aim was to really figure out what the PFDs view and their expectations and their concerns regarding biomarkers in the aspect of transplant health. So during this session, we had 3 main questions that we asked our patient partners. One was, what is a biomarker? Second is, what are the risks and benefits of using biomarkers in transplant and to assess health? And third was to investigate really how patients want to receive information about biomarkers in relation to their transplant. So insights from this discussion showed varying levels of awareness of what a biomarker actually was, and really how this is applied to their health and to figure out for transplant recipients and their overall health. Key learning points from this was that the role of biomarkers can potentially reduce the need for any invasive or more invasive procedures, such as biopsies, as Tom has mentioned. And also recognizing that the test of using biomarkers is complementary to your traditional blood work that you'd receive in clinic. We also there was an emphasis on a point that we cannot at this point rely solely on looking at a biomarker to be able to have a complete indication of your health for a transplant recipient. So in discussing the benefits of biomarkers, the conversation highlighted advantages of testing diverse biomarkers to get a more comprehensive view of your overall health, as well as into conditions such as rejection and also comorbidities that transplant recipients face, such as diabetes, and catching that earlier on. When discussing the risks, biomarkers were recognized as a snapshot of your health, but also taking into consideration that this can be misinterpreted as looking at this as one test, and it can vary from patient to patient, and it could differ as well as patients, depending on their comorbidities that they have. And it was really highlighted in the discussion that there is a strong need for a strong relationship between a patient and their physician to be able to have a really good explanation of what is a biomarker, what is the test, and depending on the result, what does that mean moving forward for your care? And lastly, when discussing how patients would prefer to hear about this information regarding their health, patients really voiced that they would like to have accessible online resources and really acknowledging that we have to cater to different informational needs. Another key takeaway was that patients really want to be able to understand what is a biomarker, to be able to say they can look at their results from clinic and really understand what that means for them moving forward in their health. So in efforts to address this in the first steps for the CXCL10 trial, we have developed and are currently finalizing some patient education brochures stemming from these conversations from our PFDs. And to provide, the overall goal is to provide basic information of what biomarkers are in relation to transplant, and hopefully to provide more information for prospective clinical trial participants, and as well, recruitment materials at each of the centers. So it was brought up that the CDTRP patient portal could be a great platform in order to disseminate this information to patients and give explanations, lay summaries, or brochures as we are creating them. And in order to keep the patient-driven process really continuous throughout this trial, we've integrated two patient partners, Sherrie and Key. This will ensure that the direction and all materials, not only this talk today, but the materials that we are creating, but the patient voice and patient partners' voice is really relevant throughout the entire duration of this project. So just to wrap up my part, the educational brochures will be disseminated to our patient partners for feedback before we disseminate them to physicians in clinic. So that will be coming soon.
Thanks, Demitra. So the way we're going to do this is that we've got two challenge questions that we're going to ask each of our panelists, and we're going to go down that way first and then back up the other way. So Sherrie gets to speak twice in a row. So the first question that we had for them was, what's the best way to engage PFDs on the participant side of the study? And I would extend that to say, what's the best way to engage participants in the process of being part of a clinical trial during the study itself? So maybe I'll start with Marie-Chantal.
Thank you for this presentation and for including me in this panel. I think there's many ways to engage patients. First of all, I think that you have made consultation with patients prior to start the study is a good way to learn about their concern, what they need to know. So that's the best thing. It was a good thing to do. And also it will depend on the patient that you engage with your study, their expertise, their interest in the study, how they want to be engaged. I think you already engage patients from the beginning of the study, which is a good thing. And also, since it's a pediatric study, I think it's important also to include young participants and children. Well, not babies, but, adolescents have a way to talk. You mentioned that you're developing brochure, but also if you want to have the buy-in from the adolescents, I have 2 adolescents at home. So a YouTube short could be a good idea to, I think, to really explain what are biomarkers and what is the study. So, and also as a, when you want to engage with patients, it's always the same principle: take time, do not use jargon, involve them from the beginning. And well, some of the general advice that you have to do for engagement, but do not forget adolescents. I think they have a word to say, and YouTube short, I don't know what it is, but it seems to be really popular.
Kifan?
Those are great points. And I just want to touch base on like the adolescents part. So being able to engage both the parent side as well as the pediatric patient side is vastly important. And then tailoring your material to what the patient and their family wants to understand. For example, I was the kind of person who started reading scientific literature review as I was in the pediatric care. So there are like people like myself who will be like, let's go jargon and let's go immunology, and which is why I'm doing a PhD actually right now in immunology. So don't shy away in these kind of difficult and certainly challenging conversations, but also create the space for that discussion. Thank you.
Thanks, Héloïse.
Is that working? Yes. Okay. So I agree with everything that Marie-Chantal said. I would suggest, Insta maybe or TikTok instead of YouTube, if you want to reach teenagers. But to be serious, when we discussed a bit about this issue before this session, and so I'm a transplant nephrologist. I've been involved in the direction of biobank at my center. And one of the aspects that I find particularly sensitive is the fact that our patients want to be good patients. They want to have that, therapeutic alliance with us. And sometimes I feel that the issue of enrolling patients in the biobank, it's not that hard because it's an observational study. But here we're talking about a randomized controlled trial, which could have different outcomes. Of course, there's equipoise, otherwise we would not do the study. But what I'd like to raise as a point of discussion is how our patient partners here on the panel or in the room can help us to find good and relevant ways to enroll patients in these trials, but with a specific attention for them not to feel pushed to enter the trial, or to make sure that they'll still feel that we're going to take as good care if they refuse. So to prevent like undue pressure to be felt by the participants that we'd like to enroll in these trials.
Great, thanks.
I'm so glad I went after you. I actually was going to speak to that. So I have seen from young pediatric, because I'm a caregiver, young pediatric to adolescents, teenager, and almost, heading towards transitioning. So hopefully what I can provide is a little bit of a breath from that continuum throughout. So early in transplant, when you come out, your role with your caregiver and your transplant team changes throughout the whole journey. So before transplant, you're just trying to gather as much data as you can, and you really hold on to every single word for word that your transplant team tells you because you're in survival mode and just trying to keep your child alive. After the transplant, you're so grateful that you actually got this second chance. So you transition mentally into a different place, a different role with your caregiver, because you're just so thankful for everything they've done for you, and you will do anything they ask you to do because they gave you your child back. So pretty much. And so it is in a unique relationship where there can be a lot of influence from your care provider into the decisions you make, the studies you get in, your life choices, and the medical directions that you head in. So I think that it's important to be very cautious with parents at that stage, because we will do just about anything you say, because you are, you've saved our child's life. And then the other piece of it is that your child's been through so much trauma at this point that you want to also protect them from any further trauma, even if it's a blood poke sometimes, because it's all trauma that links back to the transplant. Every time you go in for blood work, you remember holding your child to get that blood work when they needed it 3 times a day before transplant. So I just think that, you know, like you're trying your best to please, you know, your care team, and because you're so grateful for the care you've received and your child. And then at the same time, you feel your child's burden of the trauma they experience, like I've never experienced before. So I think you're holding all of that in the same place when you're asked to do studies. So something to consider. Like, I don't know what the right answer for that is. I think it's important to like tread lightly, provide lots of content ahead of time, and maybe, you know, on several different occasions to talk to patients about it so they can savor and truly make a decision that resonates with their family instead of a reflex decision of saying yes and then dreading the idea on the drive home and how it impacts your child. As you move through more of the adolescent or the young teenage years, they tend to say, I want to just live life. Well, mine does anyway. I want to live life, and let's make all the health stuff convenient around my schedule because I'm too busy with friends and I've got school and I've got sports. And so there's competing demands. And so I think that as well, they want to understand things that they're getting involved in. So they want a bit of a deeper understanding instead of just like relying on everything. They also rely on their care team, but I think they also have a lot more questions they want answered. And you have to grow that voice of those questions. I'm sorry, am I taking too long? Okay, sorry. But you have to really grow their ability to ask questions because they will transition on to adults, and they have to ask those questions and they have to be part of that team and decision making. So I love the different multimedia that you talked about and delivering content to them. And because maybe they won't be immunologists, but that if they are, that's great. But if they're not, they should at least have a role within their health and ask those questions. So I think the tailoring the information has to come from both their side and parent side. But the one final thing that I want to say, as you get further along as a parent in this process, you become a massive advocate for your child. So your relationship with your care team is important, but you can push also. You don't feel like you know that you're going to disrupt the boat or anything because you have developed such an incredible relationship with your care team. And you can actually, like, it's a give and take. You can really push to say, ask them questions and not feel like that gratitude that you had at the very beginning is like a stronghold on those kind of conversations. So I think that there's various ways that you need to engage. There's some considerations you have to have with patients. The final thing I'm going to say is that similar to health systems is that it's nice to have evaluative feedback throughout the process. So like even asking how the beginning engagement process was, and, you know, building up some of those, like whatever you say is anonymized, or we want your feedback. If there's things we can do, just really using the language that supports honest and authentic feedback. But I think throughout each stage of the process, it's nice if you keep a patient engaged by asking their feedback, they know they're contributing to the study.
So to actually collect that information as part of the study,
Mhm.
you know, to periodically get in touch with them and ask
Yeah.
how it's going.
Yeah, and then you have an opportunity to report back to them on the findings and how the study's going. You have
Yeah.
those sort of.
How other people think it's going.
Mhm.
Yeah. I might take the prerogative of the moderator, since we have a bit of time. Before we switch to the second question, I might just see if there's questions from the audience for the panel around this one, because we're going to switch topics a little bit. And there's been some really rich discussion here, and I just want to capitalize on that to see if anyone else has any thoughts. If you have a question, just raise your hand and we'll get you a microphone. There we go, Sean.
Hello?
Yeah, yeah.
Believe it or not, I was actually a pediatric patient myself at one time, so it's decades ago, but it's still quasi-relevant. But I would say one thing to make sure, especially the adolescents, but even the younger kids, are understanding is that you are going to help other kids that look like you. We hope to do less biopsies in the future if these tests work. So, you know, frame them in how they're helping future them. And also other kids that are going to look like them, and maybe avoid, maybe not the blood poke, but they might avoid the biopsy poke a few more times throughout their lifetime. And I'd also be curious Tom, especially with the Winnipeg experience, would there be potential for protocol biopsies and things like that? Because I mean, the Winnipeg program really was the king of protocol biopsy because of David there. But would there be appetite in the future to reduce pokes in pediatrics if these tests are successful?
Yeah, no, I mean, let me just answer that question directly. It's a long road to get there, but I think that's where we want to head, obviously, is to do fewer sort of random biopsies, if you will, and to do them only when we think we really need to. And maybe one day treat based on the results of the biomarkers rather than actually needing to do a biopsy in many cases, but that's still a ways away. Anna?
Yeah, Anna Konvalinka from Toronto. Very interesting and relevant discussion. I'm curious, Tom, if given that you've probably seen and read the trial from Europe, which is very similar to Julie Ho's trial on CXCL10 in adults, that's come out negative, but the study design is somewhat different and very confusing. I'm wondering whether you can take advantage of this first feasibility trial to maybe inform what you can learn and how you're going to design the ultimate trial. In other words, you know, what's the simplest and best way to use urine CXCL10 in terms of intervention and subsequent management?
Yeah, and you know, we can talk later about the challenges with that trial that was reported, but I mean, I think the point is using the feasibility study to inform how to better do the trial, and that speaks to both how the trial is designed, but it also speaks to what outcomes we're looking at. We will have preliminary data from this trial to make sure that the outcomes that we're targeting, that there's enough of a signal that the number of patients, for example, that we need to enroll to get there is feasible for sure. But maybe I'll ask the panel whether you have any comments on, you know, the uncertainty of scientific results, and, you know, just because we think it's going to work, is it actually going to work? How do we communicate that to patients who are participating in a trial and are part of that endeavor? Because as you said, equipoise means that I know this test detects rejection. I'm not sure that it's going to change the outcome. I think it will. That's why we're doing the trial. But how do we communicate that in a clear way that's not sort of science waffly kind of stuff?
I think the input of patient is really important because, well, we have as physicians, we are used to deal with uncertainty, but you know, when you're on the other side, you're a patient, the uncertainty is different, and you could feel the uncertainty completely. You know, one chance over 100 for a physician, it's low, but for a patient, that could be completely different. So I think how to framework the communication strategy about the uncertainty, we need, you know, the patient's voice and, you know, kids from all different age, but because that's true, they have a different understanding as they grow up and they have different perspectives. I don't know.
Yeah, I would just say like the uncertainty is a tricky thing. I think everybody has a different tolerance with uncertainty. I know from, you know, studies that we've been enrolled in, and the availability of participants is, you know, who actually enrolls of those are quite small. From some of the ones, it depends on the risk that's being taken or potential risk. So I think the most important thing is communicated several times that there's uncertainty so that patients actually hear that. Because sometimes if you just go over it once, it's not something that sticks, but I would do that on every conversation you have in regards to the lead up to this project. But also, like, it's important when you're weighing in the pros and cons to doing a study, that has to be part of the conversation at home when parents are having those. And there's often questions that parents will have. So you need to make space for those type of questions that come out of that preliminary discussion in the clinic, and then at home, and then back to the clinic.
Thank you. So I'm going to move on to the second challenge question that we had now, which is really more around once, you know, towards the end of the trial or after the trial is finished, how do we communicate the results of those of the trial? And there's the traditional sort of presented at a conference type of stuff, but we're really more interested here in how do we engage our patients and our PFD community to help us to communicate the results to our patient population. And this is potentially also going to inform a larger pool of patients who might participate in a subsequent trial. So maybe I'll start with Héloïse this time. Can I start with you?
Yes, but I
Okay.
thought you wanted.
I'm just switching.
Okay. Yeah, like full of surprises. So, so to me, one important item here is to improve the communication that each transplant center has with his patients. So, I do transplant clinic on a regular basis, and oftentimes patients have told me, like, why don't you guys have a website where we could, see what are the current recommendations for COVID vaccination, which are changing all the time, what to do if I have this, FAQs. And for instance, when you were talking about, the CDTRP website being a very nice platform to disseminate the research results, I fully agree. But how many patients, except the very motivated group that we have here, know about the CDTRP? In our general transplant clinics, I would not think it is a lot. So I think that maybe it is the role of each transplant center to have a way to communicate the existence of CDTRP or research results in which they are involved. So, for instance, at my center, I believe such a thing could be created, and you could have links to the CDTRP portal and the patient portal on the CDTRP, which is pretty well done. And you could also have, for instance, when I'm thinking about the biobank that I am directing, there's lots of research that come out of this biobank. But patients, they don't know they participated, they provided their biomaterial and accepted that we would go into their charts to review the data. And so it's because of them we can actually do that research. So they need to know, or they need to be informed. So I think that one good way, of course, is through website. We also have to think maybe about our older patients, sometimes who may not be keen or have the reflex to go on the web. But I believe, our patient partners here can help us find some solutions. But this is one of the things I was thinking about.
Thanks, Sherrie. Sherrie?
Okay, so I also agree about the website. I think you have the relationship with your transplant center, so that's your trusted relationship. And so it's nice to have the information reflected on a place that you go, or you would go, or can have access to on a platform that is your contact, your credible contact. I think within the community, there's a lot of opportunity, social media, Facebook, groups that can be a great way for patients to talk to patients. However, the one thing that I often find is that between transplant centers, if there's not consistency between transplant centers, how they disseminate to their patients, there's always like, oh, well, this center does it this way, this center does it that way. And it kind of causes confusion between centers unless there's like a cohesive plan across the centers in delivering the information and the type of information. But one other thought would be like using like some kind of like photo booth for kids to have participation in helping in that knowledge mobilization and their role. And if they have insight into how it has affected their life, it would be nice to add that personal meaning to it as well.
Yeah, this is a great question. I think it's in all aspects of scientific communication. And my take on it is, in terms of like any sort of educational material, there should be like multiple ways of disseminating it, whether like throughout the ages, whether it's something like a diagram or a play or a song, or like through YouTube videos or a website, like that's different age group. And I think there should be a conversation with the pediatric population, not just with the parents. So having like, the example of explain in 5 different levels that you see on the YouTube wired or something, that you have different levels of explanation at the kindergarten level, at the high school level, college, etc. And that's key. And another thing that's really critical especially we talked about in academic, but not in terms of like knowledge, I think is needed is that there's no stupid question. There is no judgment in anything that you ask. So if a patient or if their family member come up to you and ask basic question or something they saw on Google, you can't be like, oh, it's just WebMD, like, Google, but like, that's where we're all getting information from. That's like the first thing you even search, like you search through Google to find your literature, and other people use like search engines to find information. So having a safe space for those kind of discussions of how to evaluate information is crucial in order for patients and their family to feel trusted in having that open communication with their care provider, which is essential in building that trust and that bridge. Thanks.
That's a follow-up question, because, old people like me aren't very good at YouTube and TikTok and Instagram. I theoretically have an account, but I've got 2 followers who are both of my kids. So I mean, in terms of developing these, like where do we go to find those people who can fill that social media space for us? Because I'm probably not going to be the best person I'm not relatable necessarily to a 5-year-old. So maybe we want to find some young people who they can relate to. What do you think?
I think that this is quite a good acknowledgement. So we're often like very biased in what we know. So I think that's great. In terms of finding people that are having that ability, I think right now like CDTRP, there's a lot of trainees, right? And then that can be a bridge between trainees and young PFD, even as Sherrie has noted, they're all patient partners, even parents who are in this role, who knows what their children or their population wants. So there is resources, definitely. And, we all love to learn. And there's also resources on YouTube for how to build a YouTube channel as well. You gotta like explain it. And yeah, so I think having that space, if you give people the space, even PFDs themselves may step up themselves to fill in and have kind of like trial and error, and like everything is iterative. So you like the first time you do something is not going to be the best, and that's not there's nothing to worry about with that. Know like that whatever you put out, be able to get criticism or like feedback is essential in building a great product.
Thanks. Marie-Chantal?
I agree with everything that was said before. I just want to add also, first, I think, and I'm convinced that patients are the best to translate in lay terms, the results of studies. And also we have to, when we talk about knowledge mobilization, we have also to think about participants of the study. Many times patients told me, I participated to the study and I didn't know the outcomes. What are the results? Well, now my research team send the paper when we publish the paper to the participants, but I think that's not a good way to do it. So I think probably see with patients and particularly with, parents, kids, adolescents, how we could inform them of the results of the study. I think that could be a good way with different platforms. And finally, I wanted to say, sorry, I lost my idea. Well, also, I think to knowledge mobilization, it would be good also to look with patient partners and family caregivers to see how we could, transmit the knowledge, the results to their communities, because I know that there are a lot of patients have Facebook private groups or there are groups of cystic fibrosis patients, but as physicians, we are not aware, we don't have any access to these groups. But I think that that's a good way also to transmit information, to disseminate knowledge from studies. So again, patient, family, and caregivers are a good way to have access to these communities.
Asking the participants in the studies themselves to both give feedback about the study, but also to, interpret even what they got from the study, even if it's not the scientific interpretation.
I think that's a great idea.
Yeah.
And also, if you reach to marginalized, communities or indigenous participants, also to know how to disseminate the information in these community in a safe and a cultural safe way. I think that's of paramount importance also to have patients on board.
A few minutes for more comments from the audience. Got some mics running around. Hands going up. Two over there.
Hello, I'm thinking that one of the ways to transfer knowledge that was not mentioned is serious gaming, especially for kids.
Gaming?
Okay. I was told I have to stand up. I think kids 8, 9 years old and kids 16, 17 years old won't receive the information the same way. But if they have to play to get the information, instead of reading a paper or going through a video, it might be more interesting for them. So there are specialists. I know it's more expensive, but serious gaming has proven in industry that it's a good way to transfer knowledge.
Wow. Frank?
Just a question. Is there funding within your study to actually implement some of those things, or is it limited? Because these are great ideas. I love all these ideas. Because again, I've been involved in, I think, 11 different research projects. I think only received final papers from 2 of them. And there was no place for me to find it. I looked online. I couldn't find out how successful it was. But for the biomarkers for heart transplant recipients, I have blood tests now. I don't have biopsies anymore. So they're using blood tests to actually send it to California, get the results back to give some indication. So there's all kinds of stuff going on, but I can't access it. And I have pretty good understanding of what people are doing. So it's just making it available. Exactly the things you're saying. We have a portal for UHN. It's all great, but there's nothing in there.
Yeah.
So it would be so good, but do you have funding to actually address some of those things?
So, I mean, the short answer is a little. I mean, we carefully craft these budgets for CIHR, and then you get your budget back and it's been cut by 35%. And unfortunately, there's no discount I can get on the kits that we budgeted for. So there are some very fixed costs that we have to buy into, and that means that some of the value added type of work that we put into the grant ends up on the cutting room floor. This is not one of those things that we want to do that with, but it does speak to a broader challenge that we have in terms of when it comes to communicating research generally to patients, that the studies themselves are often disadvantaged in terms of being able to do that well. And it's sort of, organizations like this are frankly really important to do that. But it's, even with all the smart people in this room, we still don't do it very well. And I really feel like we're just at the start of this, not anywhere close to being good at it. Do you have a question?
Bit of a comment or a suggestion. I got a chance last year to work with ASHI. They asked me to be a reviewer on their plain language abstract project. So ASHI is the HLA tissue typing community, for those who don't know. So they're talking very, very technical language. Every paper they're involved with is alphabet soup, but they insisted that many of the papers that were being submitted to this competition had to also have a plain language version. And this conversation and Key, your points in particular, bring home maybe there's an opportunity for a youth language version and a parent language abstract version too, because even getting the full abstract back or the full paper back is nice to see, but if it doesn't make any sense, there should be a line at the very bottom of those that just say the so what. How far is this from actually helping a patient in their future health journey and some of those things. So kind of give that so what, but also in a plain language format.
Time for one more question.
I've been said to be really quick, so I'll be really quick. I just want to share an experience because I'm hearing so many like innovative strategy that I think are really important, but there's a lot of patient, family, and donor who actually participate in the COVID Immuno project as part of Projet Laurent that we launched in 2020. And we didn't have budget for that, and we didn't have money, but we produced like a 2-pager, like really simple infographics by a research assistant that we sent by email to participants. And I was so surprised to see like how many patient, donor, and family who participate in the study just email us back and said, oh my God, thank you so much. It has been amazing. It was really simple. It's basically the first time that we know what are actually the result of the research that have been published. And it was so simple. It took us like half a day, revise, and we just sent it by email. So I think a combination of really innovative strategy, but really simple strategy can be really useful too. And that we have the meaning and all the funding to do that.
I think that's a great note to end it on. So I just want to thank all of our panelists for helping me out today, and to all of you for your thoughts and contribution. And I'll just echo what Demitra said earlier, is that we are going to put the first version of these pamphlets up on the website, and we really would appreciate you looking at them, giving us some feedback, telling us what we need to change, any ideas for other types of information. I think one of the things I heard from here is actually just having like a frequently asked questions type of page that's just very basic facts that just doesn't rely on formatting, just relies on just having good information and having it available at different levels. So thanks very much to everybody.